Good morning, everyone. As you can see, I'm not Georgia, as it is in the program. Um, I'm currently working as a postdoc researcher on the DB group in the University of Twente, and I will present this proof of concept study that we hope to publish soon. And it's in collaboration with Erasmus in Rotterdam. So we are moving nowadays from a more conventional type of medicine to a more precise one where we can use patient-derived cells and to provide us with a more precision medicine for example, for the test of drugs. So I'm not spending too much time on the organ-on-chip subject, because we all know the value to simulate complex structures like tissues and microenvironmental and physiological cues of the functional organs. We are aware of all the advantages of these devices. So the premise for our study is the study of osteoarthritis. As you know, there is the need for predictive human and multi-tissue in vitro joint models, and the joint is a complex tissue harboring multiple types of tissues, like the synovial membrane, the osteochondral, the cartilage, the Hoffa's pad, the ligaments, the meniscus, etc. So our question is, how do disease triggers in each of the articular tissues, how this converge into osteoarthritis? So our project is to combine the organ-on-chip platforms with all the microphysiological cues with the real-time non-invasive invasive imaging to enable real-time detection and analysis of key molecular markers in order to address disease progression and to validate these models. So we try to incorporate bioluminescence on these chips. And why to use bioluminescence? Because this is the next generation of organ-on-chip biosensors that enables non-invasive real-time with imaging with high sensitivity. It enables also high signal-to-noise ratio compared to conventional fluorescence approaches, and it has the additional advantage of not presenting cytotoxicity or phototoxicity of the samples. Also, it has the added advantage to enable the incorporation of multicolor reporters. So our approach was to bioengineer cells with a viral envelope containing the NanoLook gene coupled with GFP. So it is capable of producing fluorescence at the same time as bioluminescence. Then insert them on a chip, on a 3D matrix, and after the substrate injection, the substrate for NanoLook, we could multicolor image our samples to a single cell level. So the chip that we use is a bone-on-chip design, a simple one, which has a imaging chamber and the channels that we can integrate the cell culture media. And for that, we tried to use 3 distinct gels with 3 distinct cross-linking processes, and also with distinct mechanical properties. So we used agarose, gelma, and fibrin, soft gel, an intermediate gel, and a hard gel. I assessed the diffusion rate, which is important in this case if we want to inject the substrate. And as you can see, yeah, in all of the three gels, the XFID-C molecule, which is even higher molecular weight than the substrate, diffuses in the time of 40 minutes. And also we did some extensive mechanical characterization to study those gels. So then the next step was to define the concentration to obtain single cell resolution. We came up with 2,000 cells, and we test viability on the distinct gels, which yeah, these gels are widely used for organ-on-chip purposes to imitate ECM-like environments. So we were not expecting a high cell death. So then the process basically was to create the viral particles on a HEC293 cell line, and then infect again 293 cell line, transduce the HEC293 cells to incorporate the nanolook gene on their genetic code. So then we sorted the green ones. We used the GFP as an isolation to isolate the positive cells from the negative cells, and then we incorporated them on the chip. We used a spectral microscope named IVIS to obtain the luminescence readouts, and also to over time sorry, I'm a little bit nervous. To capture the signal dependent on the cell number over time. And as you can see, we could image for 20 minutes. Yeah. Then we injected the chips with the substrate on the three distinct gels. Of course, this is hydrogel dependence, the diffusion, as I told you. And as you see in all of the gels, of course, as I told you, it's hydrogel dependent, so the diffusion rate it will be dependent on the gel that you use. But as you can see, we could capture bioluminescence on the three distinct gels. We selected two regions and we quantified. And for the time of 20 minutes, we could capture the bioluminescence. And I have a nice video for you to see here. This is a tile scan of one of the chips that we injected, and we imaged for 20 minutes. And as you can see, there is single cell resolution, which we then quantified for the signal to noise ratios compared to fluorescence. So as you can see, the bioluminescence shows a high signal to noise ratio compared to fluorescence. Of course, we normalize to the background noise, but as you can see by the arrows, specifically those cells are way more, even visually, more bright than the fluorescence ones. So this enables for us to see a better signal resolution on bioluminescence. So then we check for continuous imaging, and we injected the same chip at 24 hours with the substrate, and we kept that chip in culture for 1 day more, and we injected again the same chip. Of course, the cells suffered some stress during the imaging, because our microscope is not, was not equipped with an incubator, so there is some stress associated. But I can guarantee that it's the same cells, and as you can see, at 48 hours the same cells are capable of emitting bioluminescence. And apart from that, we checked whether if it was possible to image, to incorporate bioluminescence on 3D microtissues like spheroids. And yeah, you have the bright field picture here, you have the fluorescence picture, and then you can even see it at the naked eye. The bioluminescence is also visible, even with a higher signal to noise ratio. So the as I told you, this is a proof of concept study. So we are moving on to incorporate reporter cell lines using this technology, like for example reporters of inflammation and differentiation. And with this we can have multiple readouts on our chip. Specifically for osteoarthritis, we can address matrix formation by incorporating, for example, collagen reporters. And as I told you, we can also incorporate inflammatory reporters. Of course, this is a little bit complicated using stem cells, but we are optimizing protocols to transduce stem cells. So as a future, or the take-home message is that we incorporate successfully bioluminescence on chip with single cell and 3D spheroid resolution. This technology will enable for real-time on-site detection, because it's not endpoint reading compared to fluorescence, and the bioluminescence offers the high signal to noise ratios. And we could monitor multicolor bioluminescence cell reporters in a non-invasive real-time way. As I told you, we will adopt the same strategy to use mesenchymal stem cells and chondrocytes for differentiation inflammation reporters, specifically directed for the study of osteoarthritis. But of course, we can do this with for other disease models. I would like to thank my collaborators from the University of Twente and from Erasmus MC, and I'm happy to answer your questions. Thank you.
Yes, thank you for the talk, for the nice talk, and we are open for questions. Yes. Here.
Yeah, very nice talk. Helen from University of Pittsburgh.
Sorry?
So, okay, so in your results, and most of the bioluminescence signals inside the cells.
I cannot hear you very well. I think it's
Maybe you
echoing.
can repeat it again.
Okay, so in your results, and most of the signal is in the cells, right?
Most of the
Signal of
signal is in the cells. Most of the signals in the
Yeah,
cells.
it's in the cell.
like, can they be released into the matrix or released to the medium?
The release, the luminescence into
Right, right.
Yeah. We can use other kinds of luciferases that specific into the media. Yes, this luciferase is only for in-cell imaging, but we can incorporate now. For example, I'm using a Gaussian luciferase that we can take the supernatant and quantify the amount of luminescence, and also normalizing it to alkaline phosphatase. and we can quantify it that way.
Okay. Maybe there's another question. We have plenty of time, so feel free to ask questions. Okay. Maybe what kind of instrumentation did you use there? Maybe you can. What kind of instrumentation did you use to? Yes.
Yeah. We use a confocal microscope with a dedicated AMCCD camera. But of course, as you can see, it's just dependent also on the signal of the luciferase that you use. So you can see, you could see at the naked eye that we could even see the spheroid luminescence. So yeah, we use the MCCD camera to image.
Okay, yeah, because I thought you mentioned something like spectral imaging.
Yeah, we use the
maybe.
spectral imaging, but the point is that it's not, that microscope doesn't have the sufficient resolution for single cell imaging.
Okay, thank you. And yeah, are there questions now? Then maybe you can also explain us a little bit what have you planned of maybe to make online measurements or continuous measurements. You have started
Yeah, for
that.
continuous measurements on chip. Yeah.
Yeah. So this is planned, and what models do you want to do, or what?
Yeah, we can incorporate this, for example, on a cartilage on chip model, for example, to study the compression, and we can inject cytokines on the cell media to provide us with cues on and to study inflammation on chondrocytes in this case.
Okay. Okay. Yeah, there's a question now. Please.
Hey, nice talk. I'm curious. You mentioned that the emission was in the near infrared. However, you showed a photograph where it appeared to produce a glow, and I cannot tell what the color is because I'm daltonic. But which appeared to be in the visible. So, is the emission of the bioluminescence? What is the wavelength?
It's about it's about 500 nanometers.
Oh, okay, right. So in the green spectrum, I suppose. Okay,
Yeah.
So it's not near infrared.
No,
Oh, okay,
Okay.
all right, thanks.
Okay, and there was another question, Steve.
Hi, thank you. Sorry if this question is so basic.
No.
I'm just wondering if, because you also transduce the cells with your plasmids to express the luminescence, so is the end result quantifiable, or do they vary? Like, is it a quantifiable method? Like, can you quantifiable? Like, because in GFP, like your expression is very much dependent on the transfection efficiency, and for luminescence, is it the same, or?
Yeah, can you quantify to make the measurements? What you
If we can quantify
yeah,
bioluminescence.
exactly. How maybe you can explain how you can we quantify
The bioluminescence.
exactly,
Okay.
or the analyte you want to measure.
Yeah, basically we normalize to the background noise. So the bioluminescence is captured without the aid of any laser-assisted mechanism. So it's basically a camera with the light turned off. So we normalize the signal that we capture to the background noise, to the
So basically, like a plate reader can work.
It's a plate reader. Sorry.
Like I can use a plate reader to get the readout.
It's a plate.
No,
No,
question
it's
is
not
if
a
you can
plate
use
reader.
A plate reader to
No, we just take several pictures with the microscope and we normalize those pictures.
Okay, so you need your microscope to
Yeah.
quantify
What
it.
I was telling to that gentleman is that we can incorporate other type of luciferases that we can take the supernatant and quantify using a plate reader of fluorescence, for example.
Okay, so are there more questions? We have about 3 minutes left. Because I want to stay in time schedule with the other sessions. But maybe you can. Yeah. Okay, Then we move on to the next speaker, and this is